The next great discovery will be shared by the minds that bring it to light, by the organizations that can each see a part of the future, by people coming together in new ways to create a new possibility for health from the spark of imagination, to collaborate on innovative biomedical science and research. Collaboration for innovation. FNIH.
Good afternoon, everyone.
Good afternoon.
That's better. I'm Francis Collins, Director of the National Institutes of Health. I'm delighted to see you all here and to have a chance to welcome you to what I think is going to be a spectacular scientific experience over the next hour and a half or so. Before I get into that, though, I really want to thank Maria Freire and all the staff of the Foundation for NIH for their remarkable leadership. Maria has just been a spectacular leader of this foundation, and NIH depends so heavily on the partnership that we have with FNIH to be able to do all kinds of creative things. And some of them you will maybe hear about a little bit, although today's focus is going to be on three spectacular scientific presentations of three individuals who have won the Lurie Prize from FNIH. But I mostly now I want to just acknowledge, and that we will be doing a lot of that this afternoon, our guest of honor, Charlie Sanders, over here, sitting next to his wonderful wife, Ann. Charlie and Ann, I'm delighted to have a chance to see you again. Has been such an amazing 20 years that Charlie has presided as chairman of the board of the Foundation for NIH, leading this organization from its fledgling moments as a newborn baby into what you might now call somebody who's pretty impressive and eligible to vote and fight in wars and all that. So there'll be more comments made about Charlie from other speakers, and particularly from Steve Paul. But I just want to extend my personal thanks to Charlie for his incredible dedication to the enterprise. Charlie, I guess we have to start out with at least one round of applause. There will be more. So this scientific symposium, I think, is a fitting tribute to Charlie's legacy, especially as it highlights the work of these Lurie Prize winners. But I would like to now introduce Charlie's successor as chairman of the board, Dr. Stephen M. Paul, somebody I've known and considered myself a friend and colleague for probably more years than either of us will admit. Steve became officially chairman of the board this past May. As we were just briefly talking about, he has a few other things on his plate, but we are awfully glad that he's made the decision to devote his considerable talents and energies and activities of his cerebral cortex to the enterprises represented here by FNIH. He's currently president and chief executive officer at Voyager Therapeutics and a venture partner at Third Rock Ventures. That in itself would be enough to keep you busy, but he is now taking this on with a great deal of determination and passion. I can say a few things about his background. With more than 35 years of experience in neuroscience and drug discovery, he brings quite an amazing array of knowledge and insight to this position, including in the academic arena at Weill Cornell, where he was founding director of the Helen and Robert Apple Alzheimer's Disease Research Institute, and served as Burton P. and Judith B. Resnick, a distinguished professor in neurodegenerative diseases. But he was in industry also, so he brings all of these skills. 17 years at Eli Lilly and Company, where he was president of Lilly Research Laboratories. And he's an NIHer. He's one of ours. In many ways, he's coming home here today. Government, you may remember him, some of you who've been around a while, as scientific director of NIMH in the early 90s, just about the time I got here. But his time at NIH goes further back than that, held several other positions at NIMH before becoming SD. and was a clinical and research associate at NIH after finishing his undergraduate and medical training at Tulane. So it's hard to imagine somebody with a more compelling track record in terms of all of the issues and opportunities that will face him now as chairman of the board. He served on that board already for considerable time, so he knows what he's getting into. And just the same, he's been willing to do this. So please give a warm welcome to Chairman Steve Popp.
Thank you, Francis, and it is indeed an honor to have you here to celebrate with us at our first Charles A. Sanders Scientific Symposium. Now, before I tell you a little bit more about the FNIH and our honoree today, as well as introducing Victor Zhao, our Master of Ceremonies, I do want to thank Francis for two things. Among others, he's going to perform for us a little later, so we'll have an opportunity to thank him. But really, in all sincerity, for his extraordinary leadership of NIH. And, you know, these aren't easy jobs, and he's kept the bar very high with respect to scientific rigor and the whole notion of medical translational research and biomedical research. And we as an institution, NIH, but we as a country are much better off for his leadership. I also want to acknowledge and just state publicly that he has been one of the strongest advocates for the FNIH and really, you know, has fostered and catalyzed a number of our public-private partnerships. And although he spent the lion's share of his professional life in academia and in government service, he has seen the power of private-public partnerships and engaging all of the private sector, the not-for-profit, the profit sector, philanthropic and government institutions, all focused on doing really world-class biomedical research for patients around the world. Now, I also want to acknowledge my fellow FNIH directors, many of whom are here today to honor Charlie and to be here at the symposium. I don't know if John Porter made it, maybe not. He was ill, but we're sitting in the John Porter. He's in traffic, so he's coming. I notice his name on the building, and that's terrific. Again, the board members of the FNIH are so passionate about the NIH and about the foundation and about our role in biomedical research. We are also most appreciative of having Victor here today, Victor Zhao. He will be our Master of Ceremonies. And our presenters, Lurie Prize winners today, Carl Deisseroth, I think, will be joining us via link. Dr. Ruslan Medzitov is here. Dr. Jeannie Lee, is Jeannie here? Yes, there she is. There you are, Jeannie. And so we're going to have a wonderful, wonderful scientific symposium. Now, obviously today we are here to honor decades of inspired leadership by Charlie Sanders. So we affectionately call him Charlie. He's, throughout his entire career, has worked tirelessly to improve health and access to medical care and to further biomedical research around the world. He's done so in many different roles. He really embodies the whole public-private sector work that he's pioneered here at the FNIH. And in fact, it was Charlie who first saw the potential of bringing multiple parties to the table, share a common scientific goal, and who contribute their expertise and their support to achieve it. These public-private partnerships, which the foundation has been so effective at organizing and implementing, are many and varied, but I want to mention just a few this afternoon. These are prominent examples, but there are many examples. Let me start with the grand challenges in global health, which spurred creation of new tools to fight diseases that literally kill millions of people in developing countries, such as dengue, malaria, and now Zika, along with numerous follow-on projects aimed at controlling tuberculosis, HIV, AIDS, enteric diseases, and other major global health problems. The Biomarkers Consortium, which has created and executed more than 20 projects to develop biomarkers that really do help get new drugs approved by the FDA. They help drug companies establish that these drugs are working. Now, the consortium's I Spy 2 project, perhaps one of the stellar examples of this work, established a new landscape for clinical trials using precision medicine and adaptive trial design to allow multiple breast cancer treatments to be tested at the same time, simultaneously in a single trial. The Accelerating Medicines Partnership, or AMP, which is improving our understanding of diseases like Alzheimer's disease, type 2 diabetes, rheumatoid arthritis, and lupus. And Francis has been really the champion of AMP, and it's paying dividends in an extraordinary way and enhancing our ability to really select better molecular targets for drug discovery and to find, obviously, new treatments. Charlie made the FNIH the successful foundation that it is today with programs like these. And for that, Charlie, we couldn't be more grateful. Now let me, to get things rolling, let me introduce one of our guests of honor, Charlie Sanders. And I think, Charlie, I guess maybe you'll say a few things, and then we'll turn it over to Victor.
Can everybody hear me? Can you hear me all right? Yes. Good. Thank you, Steve. I particularly want to recognize my wife, Ann, who's had to put up with me all these years. My very able assistant, Lori Franklin, puts a very good face upon my persona, which is needed all the time. Let me also recognize my daughter, Trishelle, who is here with my son, Chris. And I'm certainly glad they're here to hear this, because if I tried to repeat it to them later, they would never believe it. Let me also say two things that I'd like for you to remember. One is public-private partnerships, which have been alluded to. The second thing is voluntarism. The first and foremost are the public-private partnerships, which were created by the passing of the legislation, which was largely authored by Ted Kennedy back in the late 1990s. Up until then, it was very difficult for the private sector to interact with the NIH, and since then, the floodgates have opened, and there have been tremendous areas of cooperation, which we'll be talking about today and tonight. The first area of partner was Pfizer, and Pfizer was interested in supporting the clinical research training grant, and has done so through the years, almost since we began, to the tune of about $20 million. It's something which is very much appreciated, and I think Pfizer is satisfied with the results because they keep funding it. Let me also point out that the other area of public-private partnerships are with the Gates Foundation, which Steve has mentioned, and that is that has been administered by Michael Gottlieb and Stephanie James. Michael, where are you? You're here today. You have on an elephant tie, which shouldn't be misconstrued. But this is originally a $200 million grant, which through time has grown to $400 million due to the fact that Stephanie and Michael have done such a great job in administering the grant that Gates has come back from time to time with new proposals for them to administer. They're really grants administrators par excellence and serve the finest traditions of science in doing their job. Now, in terms of voluntarism, let me point out that the board is totally a voluntary board. Many of us started in 1997. It started with Saul Schneider, who is here, with Dita Blair, myself, Paul Berg, who is a Stanford biochemist and later Nobel laureate, and Paul Montrone. Paul Montrone is a very active and successful businessman. He loves the numbers, and he's put in place the operating system financially, of which we've followed all through the years and has served us extremely well. And he had a very busy schedule, but he always was never too busy to give us the time that we needed to help. And then Pat Walsh, who is a surgeon at Johns Hopkins, famed for his prostate surgery. And he has been very, very loyal through the years, and unfortunately has resigned recently. Now, the first chairman was Benno Schmidt. Benno unfortunately died shortly after being appointed, And Paul Berg, who was appointed as acting chairman, found that the travel from California here was really too demanding. So he ended up stepping aside. And in a dinner held at Dita Blair's house one evening where I was not present, I found that I was elected chairman. I'd like to say that I'd run for it successfully, but in this day and time, that's probably not a nice thing to say. Now, I realize that I'm standing between you and the debate that's being happening this afternoon, and I'm not sure whether I'm doing you a favor or not. Let me also point out that the Biomarkers Consortium, which Steve talked about, is a remarkable achievement of David Wally and his colleagues. David had the idea that we should be able to gather academia and industry in the same room and come up with programs to identify biomarkers, which are markers for specific diseases, which can not only be useful in diagnosis, but in following the success of treatment. And the thing that was really incredible was that the proposal was that industry which would participate would not enjoy any patent protection. And I said, that'll never work. But it did, and it's due to the leadership and commitment of David and his colleagues. So David, are you here this afternoon?
Yeah.
No? Well, I'm sorry, because he really deserves a hand. Thank you, David. And then finally, in terms of private partnerships, public-private partnerships, I want to point out that the director before, the president before Maria was Amy Porter. She came in the foundation, and Amy McGuire, and found her husband John here on the board, which is a partnership that I don't think was envisioned by the legislature. But in any event, with all the things that are going on, I think the best days are still ahead of us. And the best partnerships that we've had, among others, are with Francis and Harold and Elias Zerhouni. And we would not be where we are today without their strong support. So Francis, thank you very, very much. Thank you and all your colleagues.
Okay, well, one
other thing, there's always one thing you forget. There are 3 speeches you give, the one you plan to give, the one you give, and the one you think you should have given. But I want to point out that one of the most remarkable examples of volunteerism is Marty Murphy. Marty had the idea that the clinical center was paying too much for drugs, so he began to sit at the doorsteps of the pharmaceutical companies and get them to donate drugs to the clinical center. And that, over the period of years, has run into millions of dollars in savings for the clinical center, and been a tremendous benefit. Marty, thank you very, very much.
Okay, thank you, Charlie. And now it's my honor to invite our Masters of Ceremony, Dr. Victor Zhao, up to the podium. We are honored, Victor, to have you here today. He is the president of the National Academy of Medicine, formerly the Institute of Medicine, and a longtime colleague of Charlie's. Now, Victor has had a very distinguished career in academic medicine. He is the Chancellor Emeritus and James B. Duke Professor of Medicine at Duke University, and the past president and CEO of the Duke University healthcare system. Please welcome Victor Zhao. Thank you.
Thank you, Steve, and certainly Charlie and Ann. John Potter just got here. And Francis and Marie Ferrer. Thank you so much for asking me to MC. I guess my main job is to keep everyone on time. Abby just leaned over to me and said, we're 15 minutes late. So I'm going to have to keep my remarks very short. But I have to tell you, when I was asked to do this, I said, of course I've got to do this. My good friend Charlie, we negotiated over the dates and the time. I'm supposedly over at London at this point with Wellcome Trust. But we managed to figure this one out. And of course, I just had my annual meeting, Francis, the last few days. But I won't miss it for the world. And of course, the question is where to have it. Charlie's my neighbor. So I can either have it in Durham or I can fly from London I can be here. And guess what? Today I'm at work. So thank you so much for holding it here. Charlie means a lot to all of us, but certainly to me. And having a chance to celebrate his 20 years of leadership, well, 20 leadership of the foundation, but multiple years of leadership, in all the things that he does in science, in medicine, et cetera, is just outstanding. How long have I known him? 40 years. Charlie, I don't think you and I at any age older than 60, right? In 1976, I was a mere fellow at the Mass General, and Charlie was what then called the general director, what we today would call the president of the Mass General Hospital. Now, Charlie is a famous cardiologist, did great work on cardiac catheterization, and so first time I met him as a lowly fellow, I looked up this tall Texan, and very soft-spoken, so I got to lean over, listen to him. But importantly, he was really imposing, and I was very intimidated, shaking in my pants, as they say. But he made me felt at ease very quickly, because that's the kind of person he is. He was down to earth, and everybody who knows Charlie will echo the same sentiments. Brilliant, scrupulously honest, visionary. You heard all the things he's done for FNIH. Funny, well, you just saw a little bit of that. Compassionate, and a great family man, and a real people person who put others first. Now, Charlie is a born leader, and someone who's dedicated his entire life to leadership to make the world a better place. And certainly, you heard his many different roles as a corporate executive, as a physician, as a leader, and of course, as chairman of FNIH. Always committed to excellence, always committed to science. My own relationship with Charlie has gone through many years, because in the early years, I think about him as this great clinician and a great administrator, but he always valued science. I worked in Edgar Haber's lab, and as a mere fellow, when I started presenting my work and very nervous at meetings, surprisingly, Charlie would always show up. At the international meetings and national meetings, the American Heart Association, I said, gee, the head of NIH knew actually who I was and I really existed. But I think it's his personal touch and his interest in individuals that, to me, stands out in the way that Charlie approaches everything. He's kind, interested, supportive, and certainly willing to bet on people. And that is certainly one thing I'm truly appreciative of. Now, he went on to do great things, CEO of Glaxo, Bristol Myers Squibb, and many roles, which are too many to enumerate. But I would say that over the years, when I was plowing my soil, if you will, we didn't have that much time to interact, but whenever I see him at any meetings, I certainly feel there's a long-lost friend. So I think it's totally fate that 12 years ago, I was brought to Durham, and Ruth and I, Ann and Charlie, became your neighbor, literally next door neighbor. And how lucky can I get? And of course, we have spent time at Cape Cod together and many other great opportunities. And Ruthie sends her love, by the way. And I'm just so pleased to be able to have somebody who's a good friend, a close friend, a neighbor, a wise person who can advise me over the many years. I think Charlie just told me I'm traveling too much. So I'm trying my best, Charlie, not to do as much. So I can say for everyone here, when I say we are very proud to be associated with you, Charlie, as a colleague, as personal friends, and we're here to honor you. But I have to say we're part of your family, and I really feel so much of this. I look at Marty Murphy here, his real family here. We're always extended family together. There's a cluster of people on Dover Road that gets together frequently under Charlie's great friendship. So I think it's fitting, Charlie, that we honor you today as the great leader. That's right. And it's also fitting that we have 3 outstanding scientists whose lives were touched by you, and they're here to speak this afternoon. These 3 individuals have a special connection to FNIH. As you heard, they're all Lurie Prize award winners. So many of you know this prize was established by the FNIH board member, and Lurie to acknowledge the trailblazing work of young scientists, and FNIH bestows this prize every year at ceremony in May, and I certainly attend some of them. So you hear from three brilliant scientists. First is Karl Deiseroff, who's the D.H. Chen Professor of Engineering, Bioengineering, and Psychiatry and Behavioral Sciences at Stanford, and Howard Hughes Investigator. He won in 2015 the Deiseroff Prize, but he also, as you know, had developed clarity and optogenetics as tools to study function of cells, especially neurons. You'll hear from Ruslan Metsitov, who's the David Wallace Professor of Immunology at Yale University School of Medicine, Howard Hughes Investigator as well. And he received the first, actually, Lurie Prize in 2013. And you hear about his work, so in interest of time, I won't repeat it. And of course, Jenny Lee, who's a professor of genetics and pathology at Harvard and MGH, also Howard Hughes Investigator. So without, she received that last year, in fact, the Lurie Award. So before I get started, let's talk about format. Each presenter will have 15, what do you think, less than 15 now? I'm teasing. Maybe I should talk less. 15 minutes, followed by 10 minutes of Q&A. And, you know, I ask you to, of course, and I know people in this room are not shy to raise your hands if you have any questions. So without further ado, let me just ask Karl Deiseroff, the first presenter, who unfortunately can't be here today in person, but is joining us by phone and by this link. Karl?
Thank you, Victor. It's a real honor to be here virtually. I had a wonderful experience at the Lurie Prize ceremony a couple years ago, and the performance by Francis Collins was by far the best part. He sang a song about what I'll tell you about shortly, and that was tremendous, but also the entire FNIH organization, including the directors, Maria Freire, everybody who's involved, just a tremendous organization, and it was a real honor to be involved and to receive the prize. I want to tell you about the work that we did that led to the prize, and also, most importantly, where we're going in the future. There's a lot of excitement in this field. Now, we work on optical technologies for probing the brain, and one of the amazing facts about this field is I'm a psychiatrist, but also a neuroscientist, and if you look back to the very earliest medical model depictions of psychiatric disease, including in 1809, this treatise by John Haslam, where he described madness and melancholy, what we would call today schizophrenia and depression, and he described anhedonia, what we would call it today, but he wrote instead, they therefore neglect those objects and pursuits which formerly proved sources of delight and instruction. This is as far as psychiatry has even gotten today in our ability to describe the fundamental underpinnings of the severe symptom domains of depression. We don't have a deeper causal understanding of how these devastating symptoms arise. And what the story that I want to tell you about today has taught us is that surprising, unexpected results from basic science, including from microbial organisms like those you see here, linked through optics and neuroscience, have allowed us to make headway on these age-old questions. Now, electrical and magnetic interventions in the nervous system are wonderful. They're fast. You can stimulate with electricity or magnetism, but these modalities of energy delivery can't distinguish different kinds of cells, and the brain is set up so that different kinds of cells that may do completely different things are right next to each other. What you see on the right is the basic principle of optogenetics is we confer light sensitivity onto targeted kinds of neurons by introducing into them light-activated proteins that move ions across membranes. That's electricity, that's the language of the brain, but it lets us turn cells on or off using light. And our allies in this endeavor, as I mentioned, are unexpected. Single-celled algae that come from freshwater ponds, archaebacteria, ancient forms of life from niches that nothing else can live in, high salt content lakes in the Sahara Desert. And we take proteins from these organisms and we put them into neurons. What you can see here is a mammalian neuron. It's about 25 micrometers in diameter across the center part, the cell body, but it has an amazing complexity in structure. And what we've put in is one of these proteins from one of these microorganisms coupled to yellow fluorescent protein, and it goes to the membrane and goes all through the cell. What these proteins let us do, and there are many different kinds, but they let us move ions into cells with different colors of light. For example, channel rhodopsins on the left let us move positive ions into cells with blue light. Halorhodopsins on the right let us do that, move negative ions into cells with yellow light, and by this means we can turn neurons on or off. We can even tune the colors. We can go beyond red and yellow. We found from this multicellular green algae a red light activated channel rhodopsin, and you can see in the middle on the right-hand side we can flash red light pulses and drive these little sawtooth-like deflections in the membrane potential of the cell, driving spikes or action potential. This is how neurons communicate, with these little pulses of membrane voltage. And we were able to create this red light activated version with some engineering and some adaptation from biology. We also were able to get to the high resolution crystal structure of one of these proteins, and this let us do even more advanced engineering. Once you get the crystal structure, you can modify things more or less at will, and what we were able to do is actually turn a stimulatory channel rhodopsin into an inhibitory one. At the lower left, you can see we're turning off spiking with blue light instead of driving spiking. And that's because we changed its ion flow properties from positive to negative using structure-guided engineering, getting all the way down to the angstrom level in our design. And the final slide I'll use to tell you about these proteins themselves shows an amazing surprise. On the left, this is a depiction of the electrostatics of the channel rhodopsin pore, the hole through which ions flow. And the red and the blue indicate whether it's a predicted positive or negative surface electrostatic potential inside the pore of this protein that sits in the membrane. And this on the left is the one we designed to move chloride ions and to become inhibitory. A year later, John Sputich's group in Texas found a naturally occurring chloride-conducting channel rhodopsin, and its internal structure was, as you can see, essentially identical to what we had engineered in our structure-guided quest to make a chloride-conducting channel rhodopsin. And this was a beautiful outcome. It shows that we understand the pore quite well, and it shows that nature's always gotten there first. They'll beat you every time, natural organisms. So a wonderful outcome, and it really highlights the value of basic biology. Now, what do we do with this approach? Well, we can study fundamental processes like anxiety. And what you can see here is a movie of a mouse running in what's called an elevated plus maze. The arm of the device that goes more up and down, a little bit more north-south, tilted to the upper left, is the so-called closed arm of the maze. It's got elevated walls, and the mouse prefers to spend its time there rather than going out into the other arm, the more east-west one that's very exposed. And this is apprehension in the absence of immediate threat or anxiety. But now with these blue letters on, we're turning off anxiety in this animal optogenetically. The animal is not changing its speed, but for the first time it's willing to include the open arms in its exploration. It's still going into the closed arms, but also out into the open ones as well. And we were able to discover that the brain has internal anti-anxiety pathways that are embedded within pro-anxiety and pro-fear pathways, and only using optogenetics, targeted delivery of channel rhodopsins and targeted delivery of light, were we able to discover these anti-anxiety pathways. Now, the progression of the field has been amazing. We only started doing these experiments in 2004, and you can see in this purple to blue transitioning curve the rapid and still growing exponentially number of papers per year published in optogenetics. Key milestones along the way, but you can see how far back the discovery of these microbial opsins goes, all the way back to 1971, and it shows the importance of all those decades of basic neuroscience before we got around to applying this set of proteins to neuroscience and to psychiatry. Now, that's where things were about the time the Lurie Prize was awarded in 2015. I'll give you a taste of where things are going in the future. We have amazing optics now. We can deliver very complex patterns of light using holograms. Spatial light modulators, or SLMs, basically play a hologram into the tissue, and we can conduct, like a conductor of an orchestra, we can conduct the firing of many individual cells across the brain all at once. And this is, of course, incredibly complicated, but really, as you can imagine, tremendously exciting. Now, that's the activity of the cells that we can play in. The last thing I want to tell you about is how we link this with wiring. The Lurie Prize was also awarded not just for optogenetics, but for this clarity advance that lets us see the local and global wiring of the cells we're interested in. We can turn brains transparent using some chemical engineering tricks. This is a mouse brain. We can see all the way through it. We can see all its wiring. Normally, you can't do that. Organisms have worked very hard in some cases to become transparent, but as you can see, the central nervous system, the brain, is very hard to make transparent, and that's because of light scattering. It's not absorption, it's scattering. At lipid-water interfaces, light will bounce off, photons will bounce off in seemingly random directions, and that makes the tissue opaque. But as you can see in the bottom half of the slide, we can take a mouse brain on the left and make it completely transparent. As you can see on the right, there is a mouse brain there. And we do that by setting things up using some chemical engineering methods so we can remove all the lipids and therefore remove all the light scattering. And I won't describe all the details of it, but what we essentially do is build a gel inside the brain that locks all the proteins and all the nucleic acids into place. This is the hydrogel embedding process of clarity. And then we can very powerfully remove all the lipids using strong detergents, and we end up with a transparent brain. And we can see not just structure, but we can link this with activity. So you can have a transparent brain, and you can register that anatomical data set with the activity data sets that you collected while the animal was still alive. And so you can have a brain-wide view of all activity of cells across the brain. You can see what their naturally occurring patterns of activity are. You can control them and see what's causally important. And then from the very same animal, you can collect local and global wiring information and get effectively the circuit diagram of the brain. Not just connectivity, but you can also label with molecular or nucleic acid probes in a very rich and multiplexed way and get detailed understanding of the components of the circuitry. And so the last couple slides here, what I'll show you here is our efforts now to integrate all these different data streams. So we have the optogenetic methods on the lower left where we're playing in activity patterns and we're seeing what really matters causally in behaviors, both natural, adaptive, and maladaptive disease-related behaviors. We're linking those data sets with local and global wiring so we can see the cells that are important, how do they connect across the brain. And we can link that with naturally occurring activity patterns as well and do all of these in the same subject. Now, people often ask, is there a direct clinical application? And this is the last thing I'll share with you. This is a very interesting question. I'm a psychiatrist. I still see patients. I just studied for and just barely passed my 10-year certification for a psychiatric practice. It was funny to have to be studying for a test at my advanced age, but I did pass it. But I don't actually work on clinical trials in my lab. I'm a basic scientist. But some people have been working on this, and they have been carrying out what you might call optogenetics-guided clinical trials with some pretty striking outcomes. And I'll tell you one thing briefly. Antonello Bonci, who's now at the National Institute on Drug Abuse, did a very interesting experiment in 2013. He took cocaine-addicted rats and was able to play in an activity pattern using optogenetics into the prefrontal cortex that turned off, effectively, the cocaine-seeking behavior of these cocaine-addicted rats, a really amazing result. Armed with that causal knowledge, which is incredibly important to know actually causally what matters, a Italian group, and Anto Bonci was also on this paper, carried out a clinical study using the currently available and approved transcranial magnetic stimulation, or TMS method, targeting the same homologous structure in human cocaine-addicted individuals, and had an experimental group and a control group, and found and reported that they were able to powerfully turn down cocaine-seeking behavior and cocaine consumption in these human beings. And it shows the value of coming in with causal knowledge. There's a lot of brain real estate that you could cover with a device like TMS. It's pretty much impossible unless you have causal knowledge, fundamental knowledge of the circuitry, to know where to go and what to do. But I think you'll see more and more of this optogenetics-guided clinical study. And so a very interesting principle. And so you can see this is the story I wanted to tell, where we've been, where things are going in the future. It's incredibly exciting from the basic science standpoint, but also just a powerful story that these microbes have helped us make headway on these ancient questions. And I would be remiss if I didn't say that all of this work was done by an amazing group of students and postdoctoral fellows and funded by the NIH and other sources, but a really interdisciplinary effort. And I want to thank you for your time and would be happy to take questions.
That's great. Oh, what a magnificent talk, and the science is just really literally mind. Are there questions? Yes, Rod.
Carl?
We're going to give you a...
Carl, this is Roderick Pettigrew.
Hi, Rod.
How are you doing?
Good.
Sorry you couldn't be in person, but your presentation came through loud and clear,
Great.
and your illustrations were quite impressive. The question I have is that it may be a bit early to begin to explore this, but might these tools and techniques be used to help us to begin to understand some of the complexities of mental health challenges and disorders, given that one typically has comorbidities, and it's sort of a common situation where a person who has OCD, for example, also has depression, or schizophrenia is comorbid with depression and so forth. That seems to be quite common. And are we able to begin to get some pathway towards beginning to understand the complexity of these types of illnesses?
Yeah, Rod, that's a great question. I think there's a few ways that the comorbidity question could be addressed with this sort of method. One thing we are already seeing is that a circuit dynamical property, let's say the balance of excitation and inhibition, if it is present in different circuits across the brain, will evoke different symptoms. So it's the same basic circuit property, and it could stem from the same genetics or the same combination of genetics and neuromodulation, but it manifests a different symptom in the person depending on which circuit it happens to be present in. And so you could have anhedonia or abnormal social behavior appearing because of excitation inhibition imbalances due to the same core genetic deficit, but manifested in different circuits across the brain. So that principle could, if it is borne out, could explain a lot of the important comorbidities that we see in psychiatry. Same local circuit property manifested in different circuits across the brain. Remains to be tested, but at least we can have a causal approach to answering that sort of question.
Thank you, Carl. There's one more pressing question. I'm told to get the show moving. All right. Listen, thank you very much for a wonderful talk.
Okay, thank you very much.
So I'd like to introduce today's second presenter, Ruslan Metsotov. And he won, I told you, the 2013 first Lurie Prize. And of course, many of you know that he was inspired by his mentor, Charlie Janeway, whom some of us know, and characterized a class of proteins called toll-like receptors, which recognize and facilitate the immune response to pathogens. And I can go on, but you're going to tell us about the science. So let's welcome Ruslan.
Thank you, Victor, very much. It's just a tremendous honor to be here to be part of the celebration of Dr. Sanders' legacy. And it's been really a tremendous honor for me to meet Dr. Sanders and others at the Foundation for NIH. It's such a wonderful organization, and it's been really huge honor to receive one of the Lurie Prizes and to be able to really appreciate all the really fantastic people associated with this organization. So I decided to, instead of giving an overview of our research or our field, I thought what I would do instead is to give one example of our research, our recent research, that is a relatively simple story, but it kind of spans different directions and different levels of science, from very basic to something that hopefully would be applicable soon to humans. So I'll talk about inflammation and disease, and one of the key points in understanding some of the diseases is the understanding the costs and benefits of different biological processes, different features of the biological systems, and in short, different biological traits. Every biological trait has some benefit, which is why it exists, and it has some cost. And the benefit has to be greater than the cost. Otherwise, it would not be evolutionarily stable. It has to be selected and to be persistent through evolution. The benefit has to be greater than the cost. So everything in this green triangle would be theoretically selectable. Everything down here would be selected against. But an important feature of biological traits that have very high costs, especially that's true for defenses, various types of defenses, including immune defenses, is that they can provide very high benefit because they can be life-saving. And if the benefit can be very high, then the acceptable cost can be also very high. So some of the traits would be like way over here. They can be providing life-saving capabilities, but they can come with very high cost. And from the evolutionary perspective, that's all that matters, that the benefit has to be higher than the cost. But from the patient's perspective and the physician's perspective, if you're on this right side of this plot, even if benefit outweighs the cost, this is what everyone would interpret and perceive as disease. And throughout the history of medicine, of course, people would complain about things that are over here just as much as they would complain about things that happen here. Of course, these primarily would not be genetic, and these are all evolved and normal part of our physiology. So if you have a fever, you may go to a doctor to ask for medicine to reduce fever, and in doing so, often the medicine would be eliminating symptoms that are actually reflections of defenses. And in some cases, this could actually occur at the expense of defenses. In other cases, it could be that this is an exaggerated form of defense, and then it would be fine to try to reduce it. The problem is that it's very hard to tell the difference, where it's an exaggerated defense and where it's actually essential part of defense that you don't want to interfere with. So we're very interested in biology in this right upper corner of the plot, biology that has to do with defenses that have very high benefit because they're life-saving, but they come with very high cost. And in the case of immune defenses and inflammation, the high cost is something we're all familiar with. It comes in the form of a variety of inflammatory conditions, inflammatory diseases. And the extreme case of it would be sepsis. It's an intractable condition that still kills too many people worldwide, including in the US. And it's a condition that, unfortunately, there is no guaranteed way of treatment. So we're interested in biology. In other words, one of our interests in this biology of survival. What happens here? Did we evolve some specific traits and mechanisms that would afford survival under conditions that can be life-threatening? So in that regard, so let me just very quickly give you, this is introduction to immunology, would be just one slide. There are two types of immunity that we have. And they indeed called sometimes type 1 and type 2 immunity. And the differences between them is very important. So type 1 immunity deals primarily with microorganisms, viruses, bacteria, fungi, and protozoa. And the goal of type 1 immunity is to destroy these pathogens, to get rid of them. To get rid of the pathogens. And so the target of response here are the pathogens themselves or the cells that they infected. And the consequence of the successful response would be destruction of pathogen and elimination of infected cells. For example, cells infected with a virus. The type 2 immunity, on the other hand, deals with things that are kind of seem completely unrelated to each other. They involve, for example, parasitic worms. They involve allergens, including food allergens like peanuts, and they involve various noxious chemicals. And the reason all these different types of challenges are all dealt with by the same type of immune response, type 2 immune response, is because the strategy of defense here is all the same. It's expulsion from the body, expelling something that you don't want inside your body. In the case of parasitic worms, you cannot use the same seek and destroy strategy because the collateral damage would be too high. It's like using nuclear bomb to fight street crime. It would be too much of collateral damage. So this type of strategy would not be usable. Therefore, what we use instead is expulsion. And expulsion is also used to deal with noxious substances, including allergens. And so the target of response here are the tissues that promote expulsion, such as smooth muscles and mucosal epithelium. And the goal of the response is getting rid of the parasite or noxious substance. So both of these types of responses, they provide these benefits, and they both come with a cost. The costs are very different. In the case of type 1 immunity, the cost would be immunopathology, autoimmune diseases, and at the extreme sepsis. In the case of type 2 immunity, the cost would be what we would call allergic symptoms. So all allergic symptoms are exaggerated responses that promote expulsion of something from the body. That includes sneezing, coughing, itching, vomiting, and so forth. So today I will just give you one example. We are interested in studying both types of pathologies, but today I will talk about this one and specifically about a set of very stereotypic responses that we've all experienced when we had a flu infection or particularly bad common cold infection. And these are a set of responses collectively known as sickness behaviors. These include anorexia, social withdrawal, lethargy, alteration in sleeping patterns, cessation of grooming, and suppression of sexual drive. These appear to be conserved through all the animals that's been studied, all the way at least back to insects. all of these behaviors. And they've been known, of course, for a very long time because we all experience them. We all know what they look like. We see them in our kids, for example. But the reason they exist have been not very clear, in that it wasn't clear whether these provide some kind of a benefit, whether these are part of our defense or survival strategy, or if these are unavoidable deleterious consequences of infection. And research over 40 years ago or so concluded that these are all motivated behaviors. This is not just things going terribly wrong. Every organism does it on purpose. But what that purpose is was still not clear. And I'll tell you very quickly a story that addresses one of these questions with regards to infection-induced anorexia. And we're also very interested in other aspects of sickness behaviors, particularly sleep patterns. But today I'll talk about anorexia and give you one example. So, as it happens in all animals, this is very well documented, for example, in veterinary science, but we also, of course, all know that when you're sick, you stop eating, you lose appetite. So in this case, these are laboratory mice that are given Listeria monocytogenes infection. It's a common pathogen that comes with food poisoning. And there is a very profound suppression of appetite. So this is food consumption here, this course of infection. Mice basically stop eating for a couple of days until they start recovering, and then they stop eating again. And so we asked, what is the purpose of this anorexia? Why they stop eating? Don't they need energy to fight the infection? And so what we did then, we infected mice with this pathogen, and then we insisted that they eat. And what happens then they all die. And we fed them only about 20% of daily caloric intake, so they're not stuffed with food. They're given only a fraction of what they normally would eat. And actually, the food we gave them is Abbott-Promote, which is the food given to ICU patients in many ICU units. And that food is very similar in composition to actually normal mouse chow. But this is the food that is given to it's true. In terms of the carbohydrate, protein, lipid composition, they're very, very similar. And so just insisting that they eat results in 100% mortality. And then we asked what aspect of food is responsible for that, because again, it could be carbohydrates, fats, or proteins, and we gave them separately these three components and found that protein and fat didn't play any role, and it was all due to glucose. So just giving animals glucose alone in the context of this infection result in 100% mortality. And then we asked what would happen if we do the opposite, if we block glucose utilization using a very simple molecule, which is a glucose derivative called 2-deoxyglucose. It's a molecule that cannot be metabolized, so it blocks glucose metabolism. And in that case, 100% of mice survived this infection. And the dose, this is here, control dose is LD50, which is half of the mice survive, half died. So this 100% survive, 100% die. Then we asked what would happen in a more extreme case if we induce sepsis using lipopolysaccharide, which is the strongest known inducer of inflammation. This is what typically is causing sepsis, and also in humans. So if LPS is injected in mice, it triggers very strong inflammation. And if we choose a dose that would kill, again, only half of the mice, and then we gave them food, then most of them died. And this is compared to just giving PBS, which is in same volumes and same osmolarity. And then we asked what would happen if we give them just glucose, and then 100% of them died. But most exciting thing was that if we give them 2DG, then 100% of them survived. And this is, again, a really difficult condition, sepsis and septic shock. It has multiple pathogenesis mechanisms, and so it was very exciting to see that just giving very, very simple manipulation was sufficient to rescue them. And then we asked what would happen in other types of infection. In the case of flu infection, for example. So mice again stop eating with flu, and then we insisted that they eat, and now most of them survived, which was not what we expected. We thought anorexia probably would work the same way in all types of infections. And in this case, giving them 2DG that was life-saving in bacterial inflammation was lethal. So now 100% of them died, which was very surprising. And so to summarize some of the other aspects of this study, what we found that the death from inflammation, from viral inflammation, was associated with decline in vital functions, suggesting there was a defect in the brainstem control centers for autonomic functions, whereas death from bacterial sepsis was associated with neuronal damage in midbrain area and, surprisingly, with convulsions, which were blocked by 2DG and enhanced by glucose. And PET scan study in these two conditions demonstrated that indeed glucose uptake corresponded to the brain areas that were affected. And because the mice with bacterial inflammation and sepsis died in the context of if we block anorexia, and anorexia leads to switch in metabolism to a fasting state, to ketogenesis, and we saw that there were convulsions right before the mice died, and then that suggested that maybe it has something to do with another connection between ketones and convulsions, which is that fact that ketogenic diet is used to treat epilepsy. And to make a long story short, we used two anti-epileptic drugs, valproic acid and Keppra, and valproic acid could save mice even if they're given this deadly combination of LPS and glucose, and Keppra could not. And the reason for this difference is because valproic acid, one of its effects actually mimics the effects of ketones. Biochemically, they have the same target. So that indicated that this target is what really mattered. And even if mice are prevented from fasting metabolism by glucose consumption, this drug that is also commonly used in humans to treat epileptic episodes was life-saving. So I will then finish with this summary of these two types of inflammation resulting in a completely opposite support programs from metabolism. And just to finish that, the clinical trials that have been performed to address the role of nutrition in critical illness were always mixed results. There was no indication that changing caloric intake or composition made a difference. And the reason for that is that there was no stratification by the type of infection that caused it. And so that's what we're planning to now do.
You are the only participant in the conference. Please press any key on your keypad to remain
these are
in
people
conference.
who performed this study, Andrew Wong, Sarah Huen, and Harding Luan. Andrew and Sarah are clinical fellows and Harding graduate student, and we got help from other colleagues at Yale, and this is our support. Thank you very much.
Thanks, Doc. I knew my mother was smart. I always got chicken soup and I got the flu. So questions? Yes.
Okay. It's a very fascinating talk. So great to hear how your studies intersect immunity, metabolism. And I was just intrigued to ask you. There have been a number of publications when it comes to impact of different nutrient supply for cancer metabolism and cancer growth. And at the high level, I could see some similarity between the bacterial infection and certain cancer metabolism publications that have indicated a favorable effect on ketogenic diets. So I was eager to understand, is it really the host cells, the normal cells, that are affected because there is considerable glucose metabolism in the tumor cells and the bacteria? So where does your change in diet have most effect? And if you could speculate a little bit the similarity between bacterial and neoplastic stages.
Yeah, so I think the answer would almost for sure depend on the type of cancer and the stage of the cancer and the status of the immune response to cancer. I think all of this will affect the response. I don't think there would necessarily be a general theme to that, except that it's clear that fasting state, low IGF-1 levels, lack of obesity, all of these are associated with reduced risk of cancer development and reduced rate of growth of tumors. But in terms of the cells that actually occupy tumor other than cancer cells, they undergo very dramatic metabolic changes themselves, including T cells and macrophages. And many of these metabolic changes are translated into whether these cells then try to promote or prevent immune response against cancer. But these are very early days. And again, I expect that there would be probably many important and interesting dependencies on the cancer type and tissue of origin in particular.
So I noticed that you found or observed activation in the midbrain with the seizure. And I was wondering if you could localize that activity to the early. Because quite a few years ago, we showed that this was a locus of control for generalized seizures by bilateral injections.
Yeah. So that's something that we would definitely would like to do to really figure out what area of the brain. The resolution we had so far was not sufficient to address that, but it's a very, very key point. And maybe we should talk afterwards about that to see what exactly those neurons are, and also why those neurons are different from neurons in brainstem in terms of the effect of glucose. Because with viral inflammation, as I mentioned, it was the brainstem area that was affected by a completely different mechanism. So it would definitely be very interesting to find out exactly which neurons are affected.
So related question, it would be interesting, and maybe you've got experiments planned to do this, to just look and see what happens in terms of gene expression in various parts of the brain stem, since we can do that now with increasingly small numbers of cells, sometimes single cells, and see what happens, the difference between poly-DIDC versus LPS early on before you've actually got the mouse really sick. What's happening there in terms of how the inflammatory response is having a different effect on the brainstem.
Yeah, absolutely. And that is, in fact, something we are currently doing to see if those brain areas react differently to inflammatory cytokines versus antiviral interferons. And that's currently in progress. And hopefully, together with more higher resolution analysis of which neurons are affected, not to mention potential contribution of glia, which is very likely to be critical here, we hopefully will get a better idea about what's happening.
Very nice work. The effects of glucose and 2-deoxyglucose on endotoxin-induced sepsis in animals, very striking. But of course, human sepsis is very heterogeneous, as I think you're pointing out. Are you suggesting that we might be able to sort of dissect sepsis and look at patients that may benefit from either glucose deprivation or glucose?
Yes, that's exactly what I'm suggesting, and that's exactly what we are currently preparing to do in a small clinical trial. The key here is to segregate the cause of sepsis, which is not always trivial, but there are ways with certain biomarkers, there are ways to do it. So we would need to do it first in a highly conservative way when we're 100% sure about the cause of sepsis. To see if we can then use these very simple dietary manipulations. And that's what we're planning to do.
Last question, Marty.
Thank you, Victor. When the FNIH, in its wisdom and the generosity of Anne Lurie, created the Lurie Prize, the very first winner was going to set the bar, and that bar had to be set high. Clearly, the bar was set properly. There's no question, as you, the inaugural Lurie awardee, set the bar because, in a way, Saul Snyder and that jury looked to see where you were at the nexus almost of the universe. And I have, therefore, not a question, but rather a petition, that here you are at really the fundament of understanding immunology, as well as not just nutrition, but also the whole physiology, including neurology. And my own perspective is that of cancer, with this proliferation now of immuno-oncology molecules and this staggering number of checkpoint inhibitors that are now rushing into clinical trials with insufficient patients really to care for these trials, to actually enter. We need, thanks to Charlie Sanders and the FNIH and David Woolley, biomarkers. Look, please, for these immunomarkers. We need desperately signpostings as to what directions we should go, and we're looking to the first inaugural Lurie Prize winner to show us the way.
Good job. Well, Rosalind and Marty, you just set a very high bar, but I'm sure the next speaker can reach that, and that's Jen Lee, who's the latest recipient of the Lurie Award. Her work on discovery of functions and mechanisms of long non-coding RNAs have certainly given us tremendous insight on epigenetic mechanisms. And are you going to talk about that today?
Yes.
Okay,
absolutely.
good. Come on.
Okay, but first, I'd like to join all of you in congratulating Dr. Sanders on so many decades of outstanding contributions and service to the biomedical community. And it actually means especially a lot to me because Dr. Sanders was president of the MGH, as was mentioned before, back in the 1970s. And what you may not know is that he played a major role in establishing the Department of Molecular Biology, which is where it's been based for more than 15 years now. And the department at the time was actually a rather unique model that represents a partnership between academia and industry. And so this is something public, academia and industry. It's something that Dr. Sanders has been involved with for many, many decades. Okay, so then the FNIH gave me a stern warning, and they said I had to give a talk, being that it's now 5 o'clock in the afternoon. I had to give a talk that doesn't go down into the weeds while presenting the science that led up to the Lurie Prize. And so, because it wouldn't be fair, and it would be a terrible imposition this time of day. And so what I'm going to do today is tell you more of a story than give you a data exposition, but you're, of course, welcome to ask me anything that you like about the data. So if I could have the first slide, please. Oh, I'm in control. Okay. Oops. Okay, so what I'm going to tell you about today is our work on sex chromosomes, always something good to talk about at 5 o'clock in the afternoon, and how what we call junk DNA plays a major role in controlling expression of genes on the sex chromosomes. Yes, here. Go to this. Oh, no, here, I can do it. I think I can do this. There you go. Okay, great. Thank you so much. Oh, we're there. We're there. No. Okay, here we go. So can you start the clock over again? Okay, so I have a story to tell you. Now, so how junk DNA controls expression on sex chromosomes. Oh, that's interesting. Okay, so first, let's talk about junk DNA. And is junk really something that's useless? Okay, so Susumu Ono, a great scientist, coined the term junk DNA back in the 1970s, based on work that was done before the 70s, in the 60s and 50s, in which it was shown that genome size had very little to do with genetic sophistication. And so, for example, a salamander has 15 times more genetic content than we do, and the weeds in your garden probably have 100 times more genetic material than we do. And so, ooh, this is, oh, now it's on auto. That's terrible. Can we stop that? All right, here we go. So the genetic sophistication has very little to do with genome size. And so now if you fast forward a few years to the Human Genome Project, which Dr. Collins directed back in the 1990s, we were told that the human genome, in fact, has fewer than 25,000 genes, protein coding genes. And that was a really startling number at the time because that's only about 1% to 2% of our genome. And okay, so maybe you can fix that while I'm talking. So that's only 1% to 2% of our genome. And it was especially sort of humbling because organisms like the fly and the worm has just about as many genes as we do. And so then we came up with this idea that, well, maybe what's important is not so much the protein coding genes, but the stuff, this so-called junk DNA, which is in between, so the 90% of the genome which is non-coding. And then the ENCODE project has now told us that, in fact, this junk is extremely active all the time. About 90% of this junk DNA is getting produced into some kind of long non-coding RNA. Okay, so the question is, what is the purpose of all this transcription? What is the purpose of this junk? So now, next slide. Here's where we sort of come in. So you know that sex in mammals is determined by a pair of sex chromosomes. And so humans come in two flavors. The boys are born with one X chromosome and one Y chromosome, and girls have two X chromosomes. And from the sizes of these chromosomes on the micrograph, you can probably tell that the X chromosome has a lot more genetic material than the Y chromosome. In fact, there are 1,000 genes here and only about 100 to 300 genes on the Y chromosome, depending on how you count and who you're talking about. And so to deal with this genetic imbalance between the sexes, what mammals do is we literally, and watch this, inactivate, how did that happen? Oh yeah, here we go. Inactivate one X chromosome in the female cell, so that males and females are now functionally monosomic, meaning we both have one pink chromosome in each sex. Okay, so we study how this inactivation of 1,000 genes happens in the cell. Now, X inactivation occurs randomly, so we can either inactivate your father's X chromosome, or you can inactivate your mother's X chromosome. And so you see this in action probably every day of your existence. So for example, here's a calico cat, and you probably know that calico cats are almost always girls. And that is because the co-color genes are present on the X chromosome, and so this patch of orange fur might represent inactivation of the paternal X chromosome, whereas the black patch represents inactivation of the maternal chromosome, whereas boy cats will either be orange or silver or dark. Okay, so turns out X inactivation is controlled by a single locus on the X chromosome. And many of us searched for this so-called X inactivation center back in the 1990s. And we now know that the X inactivation center is probably nowhere, that at least the core region, as we defined it using transgenes, is probably no more than about 100, say, at the outside, 200 kilobases in size. And so what that means is that a very small region here on the X chromosome can inactivate something which is more than 1,000 times its size. Okay? And then when we looked into this X inactivation center, what was really striking was the fact that there were no protein coding genes. And in fact, everything which is important in this region is made from junk DNA into long non-coding RNAs. Okay, so what that tells us is that this junk is actually really powerful. And it's so powerful that if we were to take this 100 kilobase sequence and we put it into any other chromosome, so-called non-sex chromosome or autosomes, we can inactivate that chromosome almost in its entirety. And so we can do this with the XIC on just about any chromosome as long as that sequence is inserted into that chromosome. Okay, so the key question here is then, what is this junk, right? And so this is what we've been studying for more than 15 years. And the junk makes this new-ish type of epigenetic regulator that we call long non-coding RNAs. And so in this region, we see exist and Cy-X, so the sense and antisense RNAs, which serve as sort of the yin and yang for X chromosome inactivation. This one inactivates the chromosome, and this one keeps it active. A small repeated RNA that targets an important epigenetic complex called polycomb repressive complex 2 to the X chromosome. And another non-coding RNA, JPX, which activates exist. And on the other side, we have excite, which activates Cy-X. And so you have non-coding RNAs acting on another non-coding RNA together with proteins to turn something on or off on the X chromosome. So it turns out that exist RNA is really important junk. So this is something that was cloned by Hunt Willard's lab many, many years ago. It's 17 kb, so it's really long. It's produced from the chromosome, which is going to get inactivated, and it has this unusual property of being able to almost like coat the chromosome which makes it. So it exists, as shown here in pink. Okay, so what we've been studying is how all of these long non-coding RNAs work together during X inactivation to produce that pink RNA and to coat that chromosome in cis. Okay, so one of the first things that has to happen is a chromosome counting process in which cells, male and female, have to figure out whether they actually have 1 or 2 X chromosomes. And so to do this, we're not actually counting the absolute number of X's. It's really the X to autosome ratio. So it's measuring the X chromosome relative to the total genetic content. And so everybody gets to keep 1 X chromosome active if you're diploid, like everybody in this room. And so a male cell would not inactivate any of his X chromosomes, regardless of whether he's diploid or tetraploid with 2 X chromosomes. But a female will inactivate 1 of her 2 X chromosomes if she's diploid, and then inactivate 2 of her 4 X chromosomes if she's tetraploid. Okay. So we proposed a number of years ago that this counting represents a titration between X-linked and autosomally linked factors that diffuse and then titrate each other. And so what happens in females is that because we have twice the number of X chromosomes, we'll make twice as many of these X-linked factors, and those extra X-linked factors will go and cause this X chromosome inactivation. And so as you might imagine from what I was saying before, this titration process, so we've learned that this titration process involves a non-coding RNA made from junk DNA called JPX, and that makes the X-linked factor, and that titrates a protein which is made from autosomes. Okay, so the idea here is the following, that during X inactivation, this non-coding junk gets produced, and when it crosses a certain threshold, as it will do only in females because we start out with 2 copies of the JPX transcript, we will see an eviction of this important CTCF protein from 1 X chromosome, and that results in the production of this very important non-coding RNA exists, which coats the X chromosome and causes silencing. Okay, so that's counting. Now then, once you've counted and decided you have to go through X inactivation, what happens is that you have to choose between your mother's X and your father's X for inactivation. And that's actually a really tricky process, trickier than it sounds, because one hand has to know what the other hand is doing. So this decision has to be made instantaneously and irreversibly so that we don't end up with 2 inactive or 2 active X chromosomes, both of which would be lethal to the cell. Okay, so then what we discovered together with the Herd Lab many, many years ago is that there's this very strange, but I think potentially important process that happens before choice actually takes place. And that is that usually the 2 X chromosomes do their own dance in cells. They're not really talking to each other. But right before the choice process, the 2 X chromosomes can be seen to come together. And they touch each other very briefly through the X inactivation center, so through this junk that I was telling you about. And then when they come apart again, we can recognize one as the inactive chromosome and the other one as the active X chromosome. So it turns out now that this interaction between the 2 X chromosomes is brokered by 2 non-coding RNAs in this region lying next to existence psi-X, and so this is psi-X and excite. And their role here is to bring a glue to the X inactivation center called CTCF again. So CTCF then joins the 2 X chromosomes together. And I have a cartoon here that illustrates basically how we think this happens. And so here's exists. This is the transcript that inactivates the chromosome. And prior to X inactivation, these pluripotency factors bind. These are transcription factors. They bind to psi-X and excite. And their role there is to turn on expression of these long non-coding RNAs. And then the long non-coding RNAs in turn will recruit this glue, CTCF, to the pairing center. And then what happens, so here you have 2 essentially genetically identical and epigenetically identical chromosomes. And then before they can go through the choice process, the 2 chromosomes come together. They have a chat, we think. And then when they come apart again, we envision, and this part is hypothetical, I should mention, we envision that what happens during this conversation between the two X's is that the transcription factors flip over from one chromosome to the other. And what that will do, so we have some preliminary evidence that demonstrates this, and what that does is that these things, these transcription factors, which normally turn on the long non-codings, will continue to do that, and the antisense RNA blocks exist expression, and therefore the X chromosome remains on. But then imagine on the other X chromosome, those factors have been lost, and so what happens is the long non-coding RNAs turn off, and that is what allows exist to turn on and spread throughout the X chromosome to establish the inactive X chromosome. So again, junk RNAs mediating very important events during X inactivation. Okay, then finally, an equally interesting and probably complex problem of once you've chosen a chromosome for inactivation, of course you have to somehow nucleate all of these silencing factors onto a locus within the X inactivation center. And then from the X inactivation center, those factors need to then somehow be propagated throughout one X chromosome. So this chromosome has 1,000 genes. And you want to be sure, absolutely sure, that these factors only go to the 1,000 genes on this chromosome and not on its homolog, which is supposed to remain active. Okay. So to make a long story short, because this represents years and years of work from not only our lab, but many, many other labs, so what we now know is that exist is made, and it interacts with lots of proteins to shut down the X chromosome. But before it can spread beyond the X inactivation center, so spread beyond this junk sequence that I was telling you about before and silence all the protein coding genes, it has to land on what we have defined as the nucleation site within the X inactivation center, and that's brokered by 3 critical YY1 binding sites. And what we've shown is that if you were to mutate these YY1 binding sites, exist literally just floats away from the X chromosome and never reaches the rest of the X. Okay, I'm almost done. Okay, so then from the nucleation site, we then spread that complex throughout the rest of the chromosome. And this didn't come out well, but what this is supposed to show is that exist, this is a time course analysis, and you can see exist first binding very few sites on the X chromosome, and then as X inactivation proceeds, it covers more and more of the X chromosome until the entire chromosome is covered. Okay. So the other thing that we've learned, and just this past year, is that exist is actually doing 2 things. It's not only recruiting these silencing factors through the X chromosome, but it's also repelling activating factors from the X chromosome. And so it's recruiting these chromatin modifiers like polycomb complexes, but it's also repelling critical architectural factors that shape the X chromosome, like the cohesins. Okay. So I want to tell you about that just briefly because it really illustrates how we might be able to perhaps reactivate the X chromosome for specific purposes. And so the X chromosome, when it's active, is folded into a shape that is permissive for transcription or for gene activity. And so here, for example, would be one regional domain that's called a topologically associated domain within which genes interact with each other, and there's lots of transcription. Now, on the inactive X chromosome, you don't see those same triangular blocks, and that's because the chromosome is silent. But when we delete exist and lose these when we delete exist and the architectural factors come back, you see that those domains reform on the inactive X chromosome in a way that is essentially identical to what you would see on the active X chromosome. And the genes turn back on within these restored topologically associated domains. Okay, so we've been learning more and more about how the X chromosome is inactivated, and we're getting hints as to how we might be able to reactivate the X chromosome for specific purposes. And so some of the things that we've learned from our X inactivation studies, as well as studies of others, is that long non-coding RNAs have properties that are really different from proteins. And that's really what makes them so important to study as junk elements. So one of the most important properties is that long non-coding RNAs can serve as a barcode that traffics different epigenetic complexes throughout the genome. And it has this ability to target epigenetic factors to a single location, whereas no protein can actually do that. And so then this gives us an idea that perhaps we can interfere with this targeting and treat specific diseases by inhibiting the interaction between, say, polycomb complexes and a long non-coding RNA. And so here you see, here's a long non-coding RNA, PRC2, gets attracted to the long non-coding RNA, and then once it's there, it binds to the gene of interest. And so the idea here is then we can inhibit this interaction by designing an antisense oligonucleotide to the long non-coding RNA so that polycomb can no longer recognize this transcript and to no longer turn down expression of this specific disease gene. And so we're partnering with Rana Therapeutics as well as others to use this gene upregulation technology to treat diseases such as spinal muscular atrophy and many, many others. Okay, and then the last thing is that so this knowledge of exon activation is also giving us the idea that we might be able to treat X-linked diseases in girls by restoring expression of good genes on this dormant X chromosome. And so the idea here is that we carry around a chromosome which is never utilized throughout life. But in specific disease circumstances, perhaps we can turn on just a small portion of the X chromosome or even a specific gene on the X chromosome so that it could be utilized to treat girls with, say, Rett syndrome, which is a horrible, horrible autistic disorder, as many of you know. These girls never learn to speak. They suffer from lifetime of seizures and acute respiratory distress. No treatment, no cure. But interestingly, in mice, Rett syndrome can be cured by re-expressing a good copy of MECP2 even after the onset of symptoms. And so we'd like to do that in humans. And this is just an example of something that we have been able to do, which is to reactivate this gene, another X-linked disorder gene, by combining treatment using two specific small molecules. Okay, so I'm over time, I'm sorry. So so again, we're partnering with various people to do this. And I want to thank all of our wonderful collaborators from multiple institutions, including here at the NIH, the NCATS, and our various funding sources, and of course, the FNIH for this incredible honor of being the 2016 recipient. Thank you.
Thank you. You would agree it's another spectacular science and presentation. So questions for Ginny? Yes, Ruslan.
Oh, yes. So the question is, could a similar mechanism be involved in regulation of other monoallelically expressed genes, right? And I think you're talking about pairing in particular, or
autosomal monoallelically expressed genes.
Yeah, so one interesting thing to come out of many, many studies in the last 5 to 10 years is that probably something like 10% of our genome is expressed from only one parental chromosome. So these include imprinted genes as well as many, many other genes, immune genes, often expressed only from one allele. And the idea is that when there's an infection or there's a need to activate T cells, the second allele would be called in to boost that response. And so we believe that, and it's been shown by others, this is not our work, including one of your colleagues, Richard Flavell, that pairing mechanisms may in fact be involved in choosing T cell receptors or immunoglobulin receptors and other such things. And so we do believe that these mechanisms can be extended to other epigenetically regulated loci.
Other questions? Francis?
It's interesting that CTCF turns out to be the glue, because of course CTCF is getting very famous for other reasons as being the protein that's responsible for creating these loops that are responsible probably for these topologically associated domains. So it's getting into a lot of action here. Now that you sort of have a hypothesis about how the components work, could you imagine setting up a system where you would get pairing, but you wouldn't go all the way to inactivation as a means of trying to figure out how to put chromosomes next to each other when they normally are not, and even changing the entire structure of the interface nucleus?
So that's a really great question. And so I think the role of CTCF in transchromosomal pairing in this case is very similar to its role in intrachromosomal loop formation. It's bringing together distant genetic elements to form, a pair. And so, except that in one case you're doing between chromosomes, in the other case you're doing within chromosomes. And so, in fact, we have done something along the lines of what you suggested. We can take very small pieces from this pairing center, again, all junk. You take a 3 kb sequence of junk from SciX or Excite and insert it into an autosome. And then what happens there is that the X chromosome gets attracted to this autosome. And by being attracted to the autosome, the two X's can no longer communicate with each other. And that results in a cell that can no longer go through X inactivation. So you can actually arrest X inactivation by tricking the X chromosomes to pair with somebody other than its sister, its homolog. So yes, so that we have done.
Okay, last question.
Maybe a very simple one.
Yes.
And I might have missed what you said, but EXIST shuts off the chromosome. Why doesn't it shut itself off?
That is a really good question. So I don't know the answer to it, except that I can provide this hint. So years and years ago, when we were creating transgenes, we made one in which EXIST accounted for something like 10% of the chromosome. So, and that's important because it amounts to a cytogenetic region on the chromosome that we can actually visualize when we create chromosome spreads. And so what we saw in that case, when EXIST was inserted onto an autosome, is that every other part of the autosome was coded by EXIST RNA, except for the transgenes, and the transgenes must have accounted for more than 2 megabases of the sequence. So the transgene was spared of the coding. And so I think that EXIST probably binds to just about everything else, but the site or let's put it this way, EXIST probably repels its own RNA from the body of its own locus.
Thank you so much. Thank you.
Thank you.
I'm sure you would agree that the prize committee chose very well the 3 Lurie prizes. What spectacular work that we've heard. Another round of applause. Well, I think this is truly an honor and celebration of Charlie's leadership. Thank you, Charlie, for bringing us together. Now, you can't be at an NIH and end a meeting without Francis coming up and do something, right? And what's that something? We all figured that one out, right? Okay, Francis, come on up.
Hope that's right. Well, gosh, Maria thought it would be appropriate, but maybe she was wrong, to have a little music here at the end. And so I was asked to think about what would be an appropriate song for an occasion like this. And that's a bit challenging after all this marvelous science to slip into something silly like this. But the doors are locked, so just don't even think about trying to run away. And there is a reception, and it's not too far off, so just hang in there. And in trying to figure out what kind of a song to pick, well, all these scientific presentations we've just heard from people who very well might be in the lineup for the Nobel Prize. So maybe we should have a song that steals from somebody who just did win the Nobel Prize. Oh, somebody named Zimmerman. Yeah, but actually, we're talking about Bob Dylan. And so how would one pick a song? Well, he wrote a lot of stuff, but it seems to me when we're talking about science, science is moving along very fast. Things are changing rapidly. Changing? The science is changing, and so are the times. So with apologies to Bob, who wrote wonderful poetry, I'm going to abuse his lyrics to make them somewhat fit the occasion, but not well enough to make anybody want to remember this later. And of course, there is a last line to these verses, for the times they are a-changing, and I'm sure it would go much better if I was not the only one singing those last lines. So here we go. Come all you who've gathered from far and from near to celebrate Charlie for his 20 years. He's pushed ever forward on science frontiers. For the future we hope to be saving. To Ann and to Charlie we raise a big cheer, for the times they are a-changin'. You can do better than that on the next verse. So we have to talk about how the times are a-changing and how we get there. Well, part of it's basic science. So come basic researchers whose skills are so fine. There's lots of new knowledge that we need to find. So much of the basics are still undefined. Those networks still need some arranging. The NIH Foundation can help you unwind, for the times they are a-changing. Of course, we love translational science, don't we? So come all you translators, don't be surprised as pathways to products are being devised. That valley of death, yet it can be downsized. Our models are ripe for reshaping. It's time to make everything personalized, for the times they are a-changing. We are in Washington, of course, so there has to be a political verse, but it won't mention the presidential candidates, because that would be a violation of the Hatch Act, and I wouldn't want to do that. But I can mention members of Congress if I don't call them by name. Come, senators, congressmen, please heed the call. While payoff from research can be a long haul, with your help the science can run and not crawl. While this generation you're saving, or history will judge that we all dropped the ball, for the times they are a-changing. Of course, why we're doing all this, it's not just about great science and great discoveries. We're actually trying to help people, and they're waiting for us. So we should have a verse about them too. Come all you who suffer by day and by night, who wait for the darkness to give way to light, who look to us here to have hope in their fight. It's their lives our hard work is saving. It's our hopes and dreams to make their future bright, for the times they are a-changing. So look to the future, the challenge is vast, and science solutions are needed so fast. We're bold, we're audacious, not mired in the past. The course Charlie's set is amazing. So come on, you people, let's go kick some ass, for the times they are a-changing.
Wow, that was amazing. Thank you. Thank you, everybody. Thank you for coming. Francis, I know you have to rock and roll and go entertain some students.
I do, yes.
So,
maybe even that will be involved.
thank you all very much for coming. This is very exciting. Thank you, Victor. Thank you, Steve. Thank you for our wonderful presenters. And Charlie, you're the best. So there's food outside. Let's go get it. Thank you very much.